to see this turn out. As I said, today we're going to be talking about precision fermentation, explaining to you what's going on within our organization and a little bit about some subsidy opportunities that we have coming up. So today's agenda, I would like to introduce you to the Wageningen team that's here on the call, and a very, very high overview of our organization. Then Maarten will take over and he'll explain some of the things we're doing, the capacities we have to do work on precision fermentation. I'll come back on to explain the public-private partnership model. So these two trajectories that we have, open calls for partners, they fall under this model. And then we'll tell you about the two we have in development. So Paul and Laurice will come on to tell you about their projects respectively. And then we'll end. I hope we'll have a little bit of time for questions at the end. If you have burning questions, clarification questions at any point along the way, please go ahead and drop those in the chat and I will facilitate that we try and answer those. At the end, we'll let you come on and ask your questions live if you want to. Good. So just super high over organizational. I don't assume any prior knowledge for those of you on the call. So Wageningen is an institution located in the Netherlands. We refer to ourselves as Wageningen University Research because we have 2 feet on the ground. One is an academic institution, the university, and one is more of an applied research. It's on paper a nonprofit, but it's an applied research institution. And from the user interface perspective, it feels very much like a classic CRO, contract research organization. And we are very narrowly scoped, so we focus only on life sciences, which is unusual. We don't have a business school, we don't have a law school, we actually don't even have anything that doesn't touch on life sciences in our organization. Food and bio-based research is an applied research part of the agro-food sciences group of Wageningen. Don't worry about that if it didn't say anything to you. But the goal, we are part of the nonprofit Wageningen Research, and our goal is to do societally oriented research. And we are aligned along these programs that you see. Each of these programs is represented by a program manager, and I am in particular for Proteins for Life, which is the protein-related research that we do. Good. I think I will leave the Wageningen overview at that. If you have questions or if you want to hear more, or if you're totally new to Wageningen, please reach out and I'm happy to do a bit more in depth with you. With that, I would like to just introduce you the team that's on this call so you can recognize the Wageningen faces. And I think, Maarten, since you'll be the first speaker, let's start with you.
Yeah, thank you. Thank you, Stacey. Yeah, I'll do a brief introduction. I am Maarten Verhoeven. I am a project leader and scientist within the microbial cell factories expertise group within Wageningen Food and Bio-based Research. My background is mainly in physiology, microbial physiology. I do a lot of work on microbial process optimization, but also to really help industry to push fermentation. And in this case, also a lot of focus on precision fermentation towards the market.
Thank you so much. Paul?
Thank you, Stacey. My name is Paul Vos. I'm the expertise leader of the group that's called Nutrition, Quality and Health. We focus a lot on in vitro research regarding digestion, fermentation, gut immune interactions, etc. I'm a biologist by background, and in the work that we do, we also connect as closely as we can to the group that focuses on human studies, so to make the in vitro, in vivo connection.
Thanks. And you're teamed up for the project you'll be introducing today with Mark. So let's go over to Mark.
Hello, I'm Mark Verton. I'm a scientist and project leader at WFBR. I have a biotechnology background, but also quite some knowledge on protein science, let's say colloid science. And I have about 30 years of experience actually in protein expression, so what's called precision fermentation more or less these days. So that's about it.
Thanks, Mark. Yeah, evolving names, right? Precision
Exactly.
fermentation. No, that's fine. It's still the same thing. Laurice. I'm not hearing you. No, it doesn't look like you're on mute. I'm not sure what's going on. I could hear you earlier.
Is it better
yes.
now?
Yeah,
Okay.
yeah.
So I
Yeah.
guess my microphone was not plugged properly. So my name is Maurice Pouvreau. I'm a protein scientist by training, and within Wageningen Food and Bio-based Research, I lead a small group of people really focusing on the application and increasing the use of alternative source of protein into food product, into tasty food products for the consumer. This is in very short who I am.
Yes, thank you very much. And you're working together with Julia, who is one of our professors and collaborators from the Wageningen University side. Julia, will you introduce yourself?
Yes, so I'm Julia Kepler, associate professor from the group of food process engineering, and my work is a lot about novel protein sources, extraction and functionality, including proteins from precision fermentation.
Yeah, awesome. Thank you for joining us. All right. So with that, we'll jump into content. That means, Martin, handing over to you. Can you resend the request for control?
Yeah, no problem.
Great. Thank you. Okay, over to you.
Excellent. Well, it's my great privilege to introduce you to precision fermentation at WFBR. Well, fermentation is, let me see if I can get, yeah, here we go. 1 second, go back. There we go. Food and bio-based fermentation are both the places where we as Wageningen Food and Bio-based are working on. And in this fermentation, I'll focus on food and specifically precision fermentation. But keep in mind that we keep this in the scope of the whole value chain when it comes to agriculture towards valuable products. This means that in our case, we have a very unique situation in which we constantly are being challenged and also are being looking at a fermentation-based process within the whole value chain. So we're constantly in contact with colleagues from Wageningen University, but also within the institute to really make sure that fermentation is a unit operation that brings value to the process in the best way possible. So tying it into downstream processing and upstream processing is key, and also ensuring a quality of the final product there as well. Let's see if yeah, here we go. So how do we proceed when we're looking at precision fermentation? You usually look at this in a very modular way. There's a lot of aspects involved. It can be going from the techno-economical aspects, but also how to really bring your process to an optimal situation. So we can provide our customers, our partners within the projects, within bilateral research projects, we can bring them the expertise we have on bringing fermentation and precision fermentation towards the market. This can be on many different topics. I highlighted some here. I'm not going to go into detail, but our focus here is really on using the state-of-the-art machinery for instance, parallel culturing of strains, but also looking at how we can optimize the strain stability and how we can optimize expression of, for instance, proteins with you, all the way towards translation to industrial process. And that can include things like monitoring your process using state-of-the-art spectroscopy techniques to make sure that quality is assured, but also that the final titers and yields are as optimal as they can be. So this is a lot of different modules, and I think the two PPSs already partly tie into this. And we also have a lot of history here when it comes to precision fermentation. And it started, I would say, with Pichia pastoris, but our genetic engineering capabilities here are much more diverse. And that's because we are not only focusing on food, we're also looking at bio-based, but there's a lot of crossover when it comes to techniques and when it comes to new applications as well. And with our institute focusing on the agricultural feedstocks, this can have many different crossovers in application. For precision fermentation, Pichia pastoris has been one of our main workhorses, and we've been very productive and very successful in engineering. Mark Wetten is one of the people that is a driving force in that, and he will be talking about that particular part in the PPS later on as well. We serve with PGA Pasteur, we serve a protein engineering with that protein engineering platform. We serve both the bio-based and food market. And precision fermentation, being a very much an emerging market right now, is definitely something that has been on our radar. So yeah, originally enzymes have been the main core focus when it comes to PGA, and we have been expressing a whole variety of enzymes for things like bioethanol production, but yeah, also for food application, enzymes can be very much of value. And we have been in contact with customers, and usually with PGA we can get a decent rate already by overexpressing with the platforms we have right now. Decent titers, and I think with optimization we can get quite far. Then one area of expertise that has been very interesting as well is looking at multimolar polymers of proteins, so structural proteins. You can think things like gelatins, but also collagen inspired proteins, and these can be de novo, so fully made, but also in some cases these are really mimicking what has been already found in nature, like with gelatin. Very fascinating topic, and a lot of things that have been done in the past on especially gelatins here. Then when it comes to animal-free food proteins, so precision fermentation on proteins specifically, we have a project currently going on caseins, so cheese proteins. Very challenging, very interesting. It's a project being funded publicly by the Dutch Research Council and WO, and we are working also there with industrial partners to see if we can get this to application. Then, well, previously developed already things like beta-lactoglobulin. Maybe Mark also touched upon that a bit later. In fact, we have been expressing that already at the beginning of this century, quite a while ago, and saw interesting yields. Market was probably not ready back then, but it seems to be ready now. So a very interesting, exciting times. Now looking at the heme proteins, a very interesting topic, and like I said, gelatins as well. So what do we have in terms of capabilities? When we have a strain, you obviously want to scale up, you want to do tests. Well, we have state-of-the-art equipment when it comes to culturing. We can do cultivation in small scale with bioreactor systems, which allows you to do 32 different conditions at the same time in a well plate. They are pH controlled fat batch systems, so you can do a lot of screening of fermentation conditions and really home in into the optimal condition to do your research in or your production in. We have fermentation capacity that you can actually opt in as a commercial partner. So that's a very interesting and unique program that's been performed here in the so to say, the campus of Food Valley in general. It's a shared research facility program. So if you want to have more information, the link that will be provided in the slide deck gives you information on that, and that also provides you already a direct access to some of the equipment that we have on site. So to pilot scale, very important also to get to generate material for tests. We have a pilot scale fermentation facility that has 20 to 100 liter fermenters on site, and we can use these to do a fit to produce a fit for food product. So this is not food grade. That's unfortunately not something that we can easily, but it does allow you to do a lot of application testing, and a lot of our customers use this easily available service and we can help them with producing material for application testing, also with other parties that are involved. This also ties in with downstream processing. We have quite some downstream processing capabilities that tie into this setup, and also a lot of application, and I'll come back to that later. It can be applied there. Application studies can be applied there as well. So when you want to go to ingredient, we are able to really put in a full downstream processing pipeline. Here you can see a picture of what it would entail. So it can be anything from cell disruption, protein recovery, towards the lyophilization and spray drying. So it can be very helpful for you to get small batches that in a very short time frame for testing. So this is something that we use. And also within big PPS projects, this is invaluable resources to make things possible with partners that usually would not be able to do without these facilities. So, well, with only ingredients, you're not there yet. So obviously we want food products. And here's an example overview that in a sense, this is only the beginning. Then we have only an ingredient. Then we need to go to a food. An example here given, casein, mycelium. I don't think I need to explain to a lot of you how complex and difficult this part of research is. But I think, again, with Wageningen Food and Bio-based Research having its food solidly in the ground when it comes to dairy production and all the connections we have with Wageningen University. There's a lot of already knowledge and know-how on how to do it with conventional dairy, and we can translate a lot of that easily towards precision fermentation. And we're very excited about that because it's a very much an emerging field. Yeah, then you can go for extend this towards meat alternatives. Here, a lot of expertise on plant-based proteins to restructure, to mix, extrude, shear, cell test, do 3D printing, you name it. We can use all these techniques and technologies to and incorporate precision fermented ingredients into that. And I think also here Laurice is going to touch upon that in a PPS proposal later on, that this can be a very exciting and new way of looking at precision fermented ingredients and how to incorporate it into the existing plant-based value chain. And then for dairy, I would say this is even more emerging in my experience. I think this is something that when it comes to the flavor and process conditions, blending, all these kind of factors are very exciting. And I think this is where precision fermentation can play a very interesting role as well. But we should not forget also the health aspect. This is also where we are strong have a strong, solid foundation at Wageningen Food Bio-based Research. Paul will touch upon that in one of the PPS proposals. But I think one of the very interesting parts that are currently not fully investigated yet, I would say, is how does precision fermentation affect health? And how can you use precision fermentation maybe to even increase health? So we're looking at things like digestibility, and also how available is the product? How does it tie into the microbiome? We have a wide variety of assays and experimental setups to really monitor it. We have the SHIME system that allows you to really replicate the human gut microbiota in a controlled setup, and this can help a lot with understanding how precision fermented ingredients and food has an effect on human health. Another part is how does it work when it comes to increase or decrease in inflammation. Also, allergenicity is an important factor here. So having proper bioassays to also mitigate risks on that side are crucial. And I think we really want to also focus our research on that and seeing how we can actually get to a product that from society is accepted and also has from a health perspective and nutritional perspective a proper way forward. Okay, then there's a small part there, regulatory work, definitely important. Novel food dossiers, we have experience in that, and with helping customers, with helping people, and also from the dissemination from big projects to see, well, how does this fit in the market? Challenging within the EU, but definitely something that, again, is emerging and becoming much more prevalent. So yeah, that's in a nutshell. Beyond fermentation, there's a lot of things happening. I think I've touched upon most of these. Again, if you are very interested in these kind of things, reach out, and I'm sure we can see how we can help. And I think with that, I can bring this towards the public-private partnership part. I'll stop controlling the screen, Stacey.
Thank you very much, Martin. You are well on time. So if there are questions or points for clarification for Martin, we can take a couple of those now from the group. feel welcome to raise your hand if you're feeling brave or drop that in the chat. Yes, Sunil, thank you.
Hi, thanks for the presentation, a wonderful presentation. And thanks for this organizing this meeting too. I'll just take two questions. I'll just pose two questions. First one is, what are the kind of novel screening tools that you have to enhance the efficiency of screening?
Yeah, thanks for the question. I would say our focus here is on how to minimize the number of replicate experiments that you need to do. We have obviously the ability to do things in bioreactors, but you know probably as much as I do that this is costly. There's a lot of things, a lot of different factors that you need to take into account, and it's also expensive to run a couple, and there's a limited number of replicates you can do. But with, for instance, this bioreactor system that allows you to do 32 cultures at a 1 milliliter scale or 2 milliliter scale, this has a great benefit. But still, with 32 cultures, it's not an awful lot, and so that's where we also introduce things like statistical design of experiment approaches, where we try to minimize replicates, but then also try to see if we can reach all the solutions within the solution surface space, so to say. So try to find actually using proper statistics all the different optimal and local maxima, minima in your solution space. So that helps a lot, and we are doing that quite often. And we're translating that mostly from the generic microbial process optimization. So we're looking at completely different processes that are not precision fermentation, but the same things apply to precision fermentation as well. So that's a process optimization. But in front of that, we obviously have the host, the microbial host, and that's where our expertise is more in. We focus our expertise on PIGIA. And because we see that that's a very valuable workhorse when it comes to the production of proteins, and that's where most of our expertise lie, and that's where we try to help customers. So that's the way we put our value proposition in. Does that answer your question?
Yeah, it does. Thank you.
Right.
Very quickly, one last question is,
Sure.
what are the kind of stability studies that you have carried out for casein?
I think I can better refer that to Mark. Did we do any proper some stability studies on those? Can you disclose it?
What do you mean by stability studies?
Stability of casein as in terms of its functional properties as well as the structure of casein in the culture supernatant, given that we know it's highly unstable and it's readily hydrolyzable.
I'm afraid I cannot comment on the contents of this project.
No worries. No worries.
So,
thank you.
Yeah.
Thank you.
Is that the same for you, Julia?
Sorry, I needed to unmute myself. Yeah, more or less the same.
Okay, yeah. Thank you for the question. A good question, apparently, because the answer seems to be confidential. We have time for one more, if there's a question from the group. Sunil was the representative of all of you. Great. Well, again, as I said, if there are more questions or things you don't want to ask in this public forum, feel free to reach out. I'm sure the team is happy to hear from you. So then we'll switch gears a little bit. I'm going to explain, Maarten referred a couple of times to PPS, which is a public-private samenwerking, which is the Dutch word. We also sometimes refer to PPPs, which are public-private partnerships, just that you understand that's our lingo for this particular project type. And I'm going to walk you through, not assuming any previous knowledge of this, walk you through on a high level what that means. So within our organization, we have 3 different project types. So we sometimes get deep science grants. These are funded by a body like the European Union in the case of the Horizon program, or by the Dutch Science Agency. In those cases, most of our funding comes from the science grant organization. The goal is usually to publish as much out of those projects as we possibly can, although companies sometimes affiliate with the projects, and sometimes there are things that we need to get approval for before we publish, like, for example, stability of casein, as it seems to be. But because these are science grant projects, the goal is usually to get them out into the open. At the very other side of your screen, we have the bilateral project model. This is one-to-one work that we do on request of companies. So companies might come to us with, for example, a precision fermented protein and say, hey, we want you to do some bioassays and see if there's, if there's any potential allergenicity or gut microbiota impact of this. That's a one-to-one. You ask us to do something, we do what we did, we hand over the IP, we hand over the results to you, and hopefully you say, thank you very much, we're very pleased. And that's a sort of the transaction. We're not allowed to apply subsidy to those type of projects, and we're working one-on-one so as not to enrich any particular company, as we are a nonprofit and beholden to the European state aid regulations. But you can always ask us for that, and you get a fair market rate for the work that we do. This model that we are going to talk about, we're going to give you two examples, is the mixed model. So that's why it's called public-private partnership, private sector companies together with public subsidy financing. Oftentimes we have both university and research involved in this, which is why, for example, Julia is here as one of our collaborators from the university side. And that also informs the content of the project. There can be a fundamental leg of the project, but there's also always something fairly applied. We get co-financing from ultimately from the Dutch Ministry of Agriculture, but that's administered by a body called the Top Sector Agri and Food, which we also sometimes refer to as TKI, not to confuse you, but if you hear that in our lingo, that's what we're talking about, this subsidy agency. Partners do need to co-finance this work, so partners bring in an in-cash and in-kind contribution, so that's important to know. We need to mix cash to be able to, from private sector in order to be able to apply for the subsidy. And partners help steer this project because partners are co-sponsoring the project. This is not a project where do whatever we want and you just sit back and watch. You're really expected to be engaged in helping make decisions about the project and helping to ensure that the deliverables that we define for the project are relevant and beneficial for you. And partners also have the right to, in some cases, take out IP. So the goal is not to develop IP, let's start there. The goal is to develop knowledge that's beneficial for multiple company partners across the value chain. But should we discover something that is patentable and collectively decide to patent that, there is a model for partners to either take that out and have ownership or take out a license from the project. And if that's a concern for you, if you haven't worked with Wageningen on this model before and you're sensitive to IP issues, please drop me a line. I'm happy to dig into this in a bit more detail with you and to show you what the contract looks like. So just to summarize, our public-private partnerships, it's pre-competitive, so it is essential that we have multiple companies participating in this. We can't do this in a one-to-one. We love the cross-value chain collaboration, so you don't need to be at the table with a direct competitor. It's actually really nice if you're sitting together with companies that feel complementary to you, your suppliers, your customers, etc. Goal is to translate fundamental insights into application, which is why we usually have these two pieces. It is a Dutch subsidy, and we are obliged to have at least one Dutch partner on each project, so somebody with feet on the ground in the Netherlands. But it's open to international partners with a preference for companies who do business with the Netherlands in one way or other, a supplier a relationship, a customer relationship. If you have the intention to come to the Netherlands as a market for you, most of the time this is not an issue. If you're concerned about that, just come to me and we'll talk it through, because the subsidy does allow us to have totally international partners if we have a good reason for them to be on board. Yeah, and actually, don't worry if these categories don't apply to you. In fact, it's open. If you're working in the food-based industry, this call is applicable to you. So the projects that we're going to talk about, I'll just do the specific generic information, and then we'll get back to the stuff really exciting for you, the content. So our typical project duration is between 3 and 4 years. As soon as we affiliate a PhD with a project, it needs to be 4 years because that's the timeline of a PhD in the Netherlands. Our project size typically works out to between 1 and 2.5 million euros. We don't like to go above that because the projects get a little bit unwieldy, and typically we look at between, I would say, 3 and 8 partners per project. So as I've already said, we get matching financing. We have a hard minimum for how much of the project budget needs to be in cash. So that's 25% of the total project budget. And then 25% can be in-kind contributions from partners. And our subsidy agency is very flexible on what that in-kind contribution can be. So for example, if you're bringing in materials for testing into the project, that's a perfect in-kind contribution. If you're bringing in tools, that's a perfect in-kind contribution. If you're doing own research that contributes to the project, that's a perfect in-kind contribution. You don't even need to disclose all of the results of that to the consortium as long as it's sort of parallel and you're giving high-level feedback back. And of course, all of the time you spend engaging with the project, coming to meetings, etc., that also counts as in-kind contribution. Again, this is a topic where, if you're concerned, please let me know. I'm happy to talk it through. And we're looking at a typical cash contribution per partner between 30 and 40K euros per year to participate in these projects. For SMEs, so if your company qualifies as a small medium enterprise by the EU standards, this is open to discussion. So we'll work that out together with you in order to enable you to participate. And if your question is, oh, but what level, what does that mean, negotiable? Well, I'm sorry, I can't answer that because it's very different if you're an SME that's a, one-person show that has just spun out of a university as compared to on the high end of an SME, which has completed, your Series C funding round and you've got 200 employees. That's a little bit different case. So it's really something that we have to talk out and dig into. I think we're coming to the end of my slides. I will just give you quickly the timeline. So we're here coming toward the end of June. We're in the process now of co-creating what are those subsidy projects that we want to apply for this year. We have a long list of things that popped up as topics of interest. These are the two that are particularly focused on precision fermentation that we'll go into today. I do want to say, we don't have skin in the game of needing these to be the topics. These are the ones that we heard from partners that we're interested in working on. The goal of today is to understand if these are resonating for enough partners to want to go ahead with them. So we appreciate hearing from you. Hey, if one of these topics triggered you, you think that's interesting, you might like to collaborate, we'd love to know. I also want to invite you to tell us if you think, hey, no, that's already happening somewhere, or this is not relevant, or you shouldn't be doing that. We think you should be doing something else. Please don't be shy about giving that feedback. That's actually incredibly helpful. So never feel like sending me a no or that's not a good idea is unwelcome. I love that. It's very helpful. So please, an invitation to do that. Our subsidy application is due September 1st, so we'll be working across summer with partners who show interest, co-creating the projects, defining milestones. Partners who want to be involved are welcome to give us feedback. Then we send it all off in September and we sit around crossing our fingers until early November when we get the notification of subsidy grant. This TKI subsidy has a pretty high success rate. So usually 2/3 of what we apply for is granted. So if you're accustomed to EU projects where that rate is something closer to 12 or 15%, this is a pretty achievable type of subsidy. After the subsidy grant notification, we go into contracting and we usually end up kicking off a project toward the end of Q1 or beginning of Q2 in the next year. So the two that we were talking about today, we aim to kick off April 2025 as long as everybody's lawyer is cooperative in that contracting process. Whew. Okay, that felt like a lot. Any questions? Let me check where we are on time. Oh yeah, great. So we do have time also for a couple of questions on this. If anybody has a question, feel free to raise your hand. I know this wasn't the part you came here to hear, right? Yes, Sunil. Hi. Our designated. Okay. Hi.
Thank you. I'll just ask one question to give others time. So I'm assuming that the chassis that you have with regards to Yarovia, Agaricus, Monascus are open to any partner to dip their beak into.
Oh, this is maybe less a question for me. Martin, do you want to jump in? So your question is around the IP, what are we free to transfer in terms of the organisms?
Yeah, the chassis that you have for
yeah,
expression of heterologous proteins.
Yeah, the platform. I mean, the Pichia platform is one we can license. Martin, do you want to comment on the others?
I think this is very much a case-by-case basis. Sometimes licenses are even for a certain domain, so we will need to investigate together with you if you are interested in certain particular platforms to see how we can match. We definitely are able to look at whether there's opportunities and possibilities. But indeed, a very good question and very relevant to explore, especially when we're investing in time and effort in a project. So those are the questions, and usually we already start asking even before the project started and look into and see what we can do there. So yes, there are definitely opportunities. So
thank you.
Thank you. Good question. Anyone else? Paul?
Yes, thank you, Stacy. Just a question around the private organizations. Do they have to have operations or in Holland or what?
Yeah, thank you for the question. That's not obliged. So the minimum requirement that the subsidy organization sets for us is that we have one partner per project that is located in the Netherlands. So we need one lead applicant that is a Dutch company. And we should fit the consortium together in such a way that the partners are complementary to each other. But it's not obliged to have operations in the Netherlands to partner on one of these projects. Great question. Thank you for asking. All right. I think let's move on then, and we'll share something about the content of the projects that we have envisioned. Oh, there's something in the chat. Hang on, let me just check. You're welcome, Dilek. Thank you for saying thank you. All right. So, Paul, I think that means it's time to hand over to you, if I'm correct.
Yep, Mark will actually start.
Yeah.
Mark. Okay.
I will try to take control of the screen.
There you go.
All right, so the first project we want to discuss with you is the PROFS project. Stands for Precision Fermentation Process Optimization for Food Safety. The background behind this is actually that we observed in conferences and discussions with customers that, the whole discussion in the precision fermentation world tends to be focused on titer. You know, it's obvious that people want to have an economically feasible project. So, of course, they focus on titer. But at the same time, we feel that product safety and allergenicity is a bit, not forgotten, but at least it's not such a strong topic in this field. And we believe that maybe it should it should receive some more attention, this topic. So what is actually the aim of this project is to evaluate the effect of process conditions on the product safety and allergenicity. I'll go into that a bit more. So we observed that on the internet, there is actually some anecdotal reports about people experiencing all kinds of health issues, tolerance issues, when they consume precision fermented products. Of course, on the internet, you never know what's really the value of these statements, but still, like I said, we observe that it's a bit hardly discussed, this topic. So we do feel it's probably good to dive into this. Let's assume that at least some of these reports are true. What is then the cause for these kind of adverse reactions that people experience? Well, it could be the protein itself. You're not producing really animal protein, you're producing, let's say, a protein encoded by an animal gene in a microbial host. So it's not the same as an animal protein, right? So what could happen is, for example, misfolding of the target protein could cause different responses in the human body. The post-translation modifications could be different when you produce something in a microbial host rather than in the animal. But we believe that probably the most important potential issue is contaminants. Because of the economics of the process, it's obvious that people want to spend as little budget on the downstream processing as possible. If you don't purify to 100.00% purity, there will be host-derived contaminants, and these could cause potentially adverse reactions. So this could be host-derived proteins, but also host-derived other components like lipids, polysaccharides, those kind of components. So those are actually the focus points of this project. So not so much the recombinant protein as such, but actually the other proteins, the host-derived proteins. So this is the proposed project structure. We envision a work package 1, which is the fermentation. Then work package 2 would be downstream processing to different levels of purity. And work package 3 would be to test then the safety and allergenicity. And of course, there will be a feedback response to the first work package again. So you can, let's say, adjust your parameters in the fermentation and see again the effect on the safety and allergenicity in a recursive process. So work package 1, if we dive into that, what's the aim? It's simply to understand the impact of fermentation conditions on safety and allergenicity. So we would like to vary and evaluate the effect of different fermentation conditions. Now, there's just a list here of all kinds of obvious things that we could vary. But yeah, like Stacy said, we are definitely open for co-creation. So please also think along what would be interesting according to you that we should check. But obvious things are, for example, feedstock. In the case of PICA, typically you would choose either methanol or glycerol glucose. It's well known that indeed in PICA, for example, the whole metabolism of the organism is so different that you can definitely expect quite huge differences on the spectrum of compounds in the medium. So that could then again have different effects on the allergenicity and safety of the product that you produce. Well, different media in general, there's all kinds of media that are recommended for different hosts, so you could vary there. Feeding approaches, you have the diostat, substrate limiting, substrate excess, all kinds of different feeding approaches exist for different hosts. So those are things we could evaluate. And yeah, well, general culture conditions, temperature, pH, oxygenation, factors like that. Oh, sorry. Yeah. So what would we then like to ferment? At least on our side, it's also possible that our partners would do fermentations. But talking from our side, what we would like to ferment is beta-lactoglobulin in P. capistoris. Why this particular model system? Well, to be honest, a lot of startups are very interested in beta-lactoglobulin. It's one of the hottest proteins at this point because it's the major whey protein. Quite some startups are active in that field. PICA seems very favorable or often used host system among startups. That's one reason to also use PICA. And of course, like Martin already said, we have quite a lot of experience with PICA already for like 30 years. And already in 1996, we produced beta-lactoglobulin way ahead of this whole precision fermentation thing. So we have a lot of experience with this particular protein also. And not unimportant, it's an easy protein. So the caseins were already discussed. Those are difficult proteins. Beta-lactoglobulin, yeah, if you just express it, don't do anything special, you already have a good yield. That's important for being able to indeed test some things. So we don't want to have a difficult protein at this point. Like I said, partners can definitely also bring in their own fermentations, even bring in strains if they feel like that. Bring in half products of their process if they're interested in investigating the effect on allergenicity of certain half products in their process. So we're all open to that. But it's important to keep in mind that indeed we're focusing on the host-derived contaminants, so not so much the target protein as such. So the second work package would then be the downstream processing. For the beta-lactoglobulin, we actually in the university, the group of Julia Kepler, we have developed a downstream processing system for beta-lactoglobulin. So that would allow us to indeed harvest this protein from the PICA supernatant, because it's actually, I didn't say yet, but it's a secreted protein, so we can harvest it from the PICA supernatant at various levels of purity. And then you could, of course, evaluate the effect of doing crude purification or quite decent purification or excellent purification, and evaluate a bit the effect on safety and allergenicity. Like I said, partners could bring in their own cell-free fermentation streams and purified products. I think I'm skipping one slide.
Yeah, if we can go back to this one, indeed. Thanks a lot.
Thank you. Yeah.
Do you want me to...
So actually, this is I'm actually done, Paul. So please, the floor is yours.
Thanks. Yeah, so I guess what is very important in this overall setup is to link the different aspects of processing to relatively simple tests related to safety and allergenicity. And I guess also as a first step in that, digestibility is also very important, which may actually relate also to, for instance, tolerance issues that may occur and which is difficult directly to study in an in vitro setup. So what we aim to do as a first step is take an in vitro consensus protocol for digestion, which is called the InfoGest protocol, to look at gastric phase digestion and small intestinal digestion with more pepsin and trypsin dominance of protein breakdown, and focus then, as Mark already said, mostly on host contaminants, because also aspects of, for instance, allergenicity related to differences in glycosylation or phosphorylation of proteins is also addressed in other projects. So we think that there's really added benefit to focus on the host contaminants in this project proposal. So then the interaction between host contaminants and the digestibility of the target protein could be an interesting outcome in itself, but it's also an important step for then going towards more allergenicity and safety or gut barrier related effects as a second step. And we could look at the interaction between host contaminants and the target protein, but also take, for instance, a reference protein to see whether host contaminants may have an effect on digestibility. So there's different setups that we could take here to take first steps before moving into more complex systems. If you could go to the next slide, Mark, if that works. So what we would like to do is focus on relatively simple or uncomplicated screening systems. So to not make the safety testing a target in itself, but really to have it as an outcome to inform how to do the processing steps and how to choose the optimal variants. So what we would like to do is focus on, for instance, the allergenicity aspects on especially IgE cross linking, binding and cross linking, but also IgGs, where in established allergic patients, you could take sera and look at binding to the host contaminants. I guess here it's the question of taking the right allergic target group. Depending on the host system, we would need to make the best choice here. But for instance, Baker's yeast also, since it's quite closely related to Pichia, could be a good starting point. We could do additional things also to look at, for instance, inflammatory potential of host contaminants to see whether other immune reactivities could be relevant here. We have a lot of experience in those. But another important aspect is the potential toxic or negative effects on epithelial lining cells, so which make up the first line of the small intestine and also the large intestine. And there we have a lot of experience with cell lines, either single cell cultures or co-cultures with different cell lines combined to look at epithelial cells. And there we could expose these cells to either the proteins with the different levels of contaminants before and after the digestion, and look at potential effects on the gut barrier, which we can measure quite easily, for instance, with the electrical resistance across the barrier, the TIR parameter, and there's other simple parameters as well. Optionally, but this would really depend on the specifics of the setups that we're going to test, we could also look at interaction with microbiota. As Maarten already mentioned, we have the SHIME system where we could look at these kind of effects, but also there's simpler methods looking at batch fermentations. But here it really depends on whether there's a rationale to look into potential interaction between metabolites and microbiota. If that is considered relevant, we could also look into these effects to see whether there's potential for toxic compounds to be produced. And then finally, the last point is important to look also at the characterization of the contaminants and the impurities. So both protein and non-proteinaceous impurities, where we could obviously look at the proteomics approach, but also look at based on in silico approaches on the genetic makeup of the host strains to look at in silico allergenicity screening, and take a similar approach in non-proteinaceous impurities to characterize what is there and then to link that to literature results or information, but also do more targeted biological work in vitro if there's important leads to follow up on. Last thing that I want to mention here is that there is a PPS project running, so with the similar setup, which is called SafeFerment, which actually focuses on developing methods for safety assessment. So I want to highlight here that we think that this proposal is really complementary to that project in that we want to take, more simple readouts here and to optimize the process. And not focus on the development of the safety assessments themselves. So in this way, we think that this project really adds something unique and relevant to the field. And with that, I think that was the last slide for this proposal. So then I guess it's good to hand back to Stacey and see whether there's questions on this, I assume.
Yes, thank you, Mark. Thank you, Paul. We do have time for a question or two. Come on, you guys can't rely on Sunil the whole time. Sorry. Bad joke. Any questions, comments?
Sure, Stacey. This is Sarah. I have one.
Hi, Sarah.
Mark and Paul. Hi, good to see you. Great presentation. I think a really interesting project. How are you thinking about kind of separating out the effects that kind of process could have in changes on the post-translational modifications from the other kind of background proteins or other products? Because I can imagine those could be kind of obviously co-correlated or impact each other in some of these assessments.
Yes, thank you. I think that's a great question. At least I can start an answer. Maybe Mark, you can add if you have additional ideas. So what we have discussed is that we could look at more or less the empty host and combine that with a reference protein, which could be the natural counterpart of the target protein, as well as with potentially different levels of purified, precision fermented proteins. So in that way, looking at the interaction between the contaminant materials and the protein of interest. And I guess also if there's a specific reason to look into the the protein of interest itself, we could still, I guess, consider that. But we think that there's other projects that focus on those aspects more. So we don't want to have the main focus of this proposal on the target protein itself. Does that answer your question?
It does, yeah, thank you.
Mark, you unmuted. Did you want to jump in and say anything or just agree with Paul?
I kind of agree with Paul. That's, yeah, it's a complete story.
Okay, thank you. Okay, I'm going to take, there's a question in the chat from Rita. What's the anticipated timeline from the provision of protein samples of varying purities to the conclusion of the allergenicity? Is that less than a year, 1 to 2 years? Oh, thank you, Rita. This is a question we get so often. So the total project will be, of course, a longer 3 or 4 year project, but very familiar to us that partners have shorter term deliverables that they'd like us to see us tackling up front. So I guess to you as project leader, Paul, how do you see that shaping up?
Yes, thanks. I think so the expectation is when we use ELISA-based methods, which are actually also being set up in this PPS SafeFerment project, so we could work in parallel with that, or using Western blot technologies. Those are relatively simple approaches. So I would expect that actually when the protein is there, that we could have a relatively fast assessment. So I would expect that that would be within a year's time frame rather than 2 or 3 years time.
Yeah, great. Thank you. Cindy, I'll raise your hand. Bye, Carrie. Thanks for joining.
Thank you. Very quickly, so given that you are conducting TERs, transepithelial resistance, which is great, have you considered conducting the similar kind of TER with endothelial cells, brain microvascular endothelial cells, to look at blood-brain barrier permeability, because there could be metabolites that would seep in. Of course, the concentration might be much lesser, but have you considered that?
Thank you, Sunil. That's a very interesting question. We haven't considered it, in all honesty, for this proposal. So we're really focusing, I guess, on the first steps and I think the first pertinent question on what happens in the gut. We do have experience in also looking at the combination of gut barrier and blood-brain barrier in other projects that we're working on that focus more on, I'd say, well, brain conditions. So we do have experience in combining these models. I'm not sure whether that will be feasible within this setup because it creates a lot of additional complexity in the in vitro systems. But well, I guess we can always discuss further on the project proposal or for other types of projects.
Very quickly, have you considered using S9 fractions or microsomes? Because it's great that you're doing IgG and IgE binding with the host proteins, but oftentimes the microsomes metabolize them into much different kinds of metabolites.
So, sorry, your question was on IgE binding to the microsomes, or?
No, so the microsomes would digest anything which is there in the bloodstream, right? So you would actually get a different metabolite when compared to the host protein contaminant, I mean, host contaminant protein. Mm-hmm. So you're essentially not even considering a full spectra of S9 derived metabolites or microsome derived metabolites.
I guess what we are considering now is to really look at what happens basically first with just I'd say the starting materials and after digestion, which is also already quite a complex matrix to work with. But I guess what is also technically possible is to look at the I'd say the absorption with the epithelial cells and take samples from the basolateral side, so basically the blood side. But that, I'd say, amounts will be so limited and that it will be, I think, very hard to impossible to then do the steps afterwards.
Got it.
So I think that will be the limitation of this setup.
Thank you.
Thank you guys for those really great questions. I'm happy to see you all engaging. I think in light of time, we'll stop it there. But again, the invitation to engage, I think Paul lights up when you give him hard questions. So feel welcome to do so. All right, let's shift focus a little bit over to our other public-private partnership and development. And Laurice is going to introduce that one for us.
Yeah. Did
you have to take control? Yeah, no, I didn't get a request to give it to you.
I did send it to you, so.
Oh no, I'm sorry, it's lost in cyberspace.
Yes.
You can try again, or I can just flip through for you, whatever you prefer.
It's saying that it's waiting for approval, so.
I'm
so
sorry.
I cannot.
Okay, I will.
Maybe I will try again. I will try again. one more time. No?
I'm sorry. I
Okay,
would
it's fine.
absolutely approve if I could.
Just
I
go
would
to
flip
the
for you.
next. Just go to the next slide.
Okay.
So welcome everyone. So yes, the topic is slightly different. What we would like to explore in this project, and of course, I am working on this project idea with Julia Kepler. So please, Julia, if I'm forgetting something, please step in. Yeah, you are the expert in precision fermentation. So our project is really on mixing animal-free milk protein with plant protein. And why are we interested by this topic is that what we can see and hear is that precision fermentation-based protein are still relatively expensive to produce, even so there is great work and a lot of improvement that is made in this field, but it still remains expensive for alternative food product. And on the other hand, plant protein do not really provide the same taste experience for the consumer, but also the same nutrition and functional performance as their animal counterpart. So we are looking for a win-win match there. What we would like to work on is develop hybrid product combining animal-free protein, so dairy protein from precision fermentation, and plant protein that delivers the taste and the texture experience to the consumer. So again, the consumer expectation there is at the core of this project. And what we aim to develop is a toolbox to provide mixing strategy between plant and animal-free milk protein for optimal functionality for a specific target application. So again, it can be different type of food products that you in the dairy alternative space or a different type of consumer category. It's something that we will work on and define together within this project. So you can go to the next slide. And why do we believe that we need to study this? Animal-free beta-lactoglobulin, differs in functionalities than the animal beta-lactoglobulin. For example, you can see on the right side of the screen that depending of the variant of beta-lac that you have expressed, that it can have a higher gel strength and a lower foam stability when the emulsifying capacity remains unchanged. On the other hand, a crude beta-lac from a different strain can have a lower gel strength, lower foam stability, but again, a very similar emulsifying capacity. So depending of the proteins that you're obtaining from precision fermentation, either depending of the variant that you are producing or expressing, or the purity of the ingredients, you can have different functionality. So it's very important to characterize and to understand how also this new type of protein will interact with plant protein. We believe it will give different outcome. If you can go to the next slide. What we know from mix of dairy and plant protein, that it can actually have a synergistic effect. This is an example on the emulsifying properties of whey protein by itself, pea protein by itself, or sodium caseinate, so in SC on the graph on the left side. And you can see that the pea protein and the sodium caseinate stabilize emulsion, but also show some destabilization over time, so flocculation and coalescence during storage. While if you have a blend of whey protein with PPI or PPI with sodium caseinate, they are actually forming a stable emulsion during storage for a longer period of time. So what we want to find out in this project is, do we get the same synergistic effect while working with precision fermentation proteins, dairy protein, either beta-lac or casein? To the next slide. if you are looking at a lot of the dairy food products are more acidified type of gel. So if you think of yogurt or if you think of cheese, so this is an example again on synergistic effect between sodium caseinate and plant protein in an acid gels. You can clearly see on the left side on the microscopy picture that if you are partially replacing sodium caseinate by soy protein, you have a change in macrostructure, but you have also a change in the mechanical properties of the gel on the right side, so the Young modulus. And you can see that if you are, how to say that, pre-treating the soy protein before to mix it with the sodium caseinate, or if you are pre-treating the mix of protein, you can actually recover the microstructure of the gel, but you can also have very similar mechanical properties. So again, it is a very interesting synergistic effect. Can we also, again, get these similar performances with this protein, dairy protein from precision fermentation? This is what we want to find out in this project. Next one. This is my last example. This is synergistic effect between whey and plant protein. This is again soy protein. And again, what you can see is that partially replacing really changed drastically the microstructure of the gel. So if you go from the left top picture, you have a very coarse gel, and as soon as you add soy protein, you have a completely different differences in microstructure. And this is inherent to the properties of the protein. But what we also saw is that by mixing these two proteins, we can actually have a G prime, so a stiffness of the gel which is higher than the sum of each contribution. So it is very interesting to better understand, and I think it will really open new avenue for alternative food products, so mix of dairy and plants, from dairy from precision fermentation, of a new range of product which is more appealing for the consumer. If you can go to the next slide. So this is, you will recognize some of the icon because Mark and I use the same one for consistency between our project idea. This is a project structure of the proposed project structure. So one part is on the production and the downstream processing, the work package 1. The work package 2 consists of characterization of the precision fermentation stream, protein and of different purity, and also the blend with animal protein. And the third work package is really trying to, from the learning of work package 2, try to apply this learning into model food products, which can be discussed together with you of which one has is the most interesting for you. If we can go to the next slide. So work package 1, so the production and the downstream processing. What we want to understand there is understanding the impact of protein purity. So for example, from 25 to 100% of protein purity after downstream processing on the functionality of the ingredients that you are achieving. So it's not focused on the health aspect like Mark and Paul just mentioned, sorry, Paul, just mentioned or talked about before. There we are really focusing is, do we get completely different functionality due to the minor components which are present in the stream from precision fermentation that we are obtaining. And like Mark mentioned before as well, we are either open to have our own production and do our own downstream processing using an already developed process and patented process, or to also obtain stream from the industry, from our industrial partner to actually check and characterize this fraction from a functionality and also from a flavor. I think one aspect that we also would like to touch, and I did not mention that before, is that does this new dairy protein also bring the dairy flavor? Or how does flavor added to this mixture will have actually a similar effect than the dairy flavor we are all used to? If we can go to the next slide. The work package 2 is really characterization of the protein blends. So animal-free dairy protein with plant protein, with the aim of really developing a toolbox to provide mixing strategy for our industrial partner between plant and animal-free dairy protein for optimal functionality for specific target application. So what we aim to do is characterization of protein blends and to define what is a minimal amount of this precision fermentation protein to obtain the optimal functionality in terms of flavor and in terms of techno-functional properties. We aim to use a medium to high throughput method. When applicable, it's not always applicable, to actually look at the functionality of this blend. And we would like to compare this functionality of the blend with either whey protein isolate or whey protein concentrate or caseinate. So how does this one compare to the blend with the precision fermentation protein? So far, what we foresee as functionality of interest is the gelling properties and the interfacial properties. Interfacial property will give us an idea of the emulsifying and foaming properties of this blend. At the end of work package 2, what we also would like to aim is to have a selection of interesting ratio or blends of this protein to be tested in work package 3 in different product categories. So it can be different blend for different application. If you think of cheese or beverage, it might be something different. If you can go to the next slide. So it's really about the application in model food product. It evaluates the functionality of these blends that were selected in work package 2 in a real food product. So it's not only the protein blends, but it's also with the addition of fat or other components from this model food product to look at, for example, milk or beverage, yogurt or cheese, and really looking at the texture of these final products, the sensory properties after food processing. So for example, if you think of cheese or hard cheese, what would be the melting behavior of this blend in the model food product? For the yogurt, is what would be the smoothness of the yogurt? What would be the sourness of this yogurt? And for the beverage, for example, what would be the stability upon storage? What we also foresee, because we really would like to co-create this project and to have the partner as involved as possible. For me, the most successful consortium project are really when the partner are part of the team and really participating in developing product and testing product. So what we see as a potential in-kind contribution is, of course, providing the protein from precision fermentation of the plant protein, and to really test the product concept at the partner. So for example, fermentation for yogurt. And if you are, depending on which country you are coming from, if you can perform sensory analysis of this product, it would also be of great help and support for the consortium. And I think this is my last slide. Yes. So if there is any question.
Yeah, thank you, Louise. I already see one question in the chat and it's been voted up. So let's do that one. Thank you, Martin, for the question. Is the intention to only target applications in, lots of questions, in the dairy field, or are we open to other application areas where milk products, milk powders, for example, are being used?
in principle, I think we are open to it. For me, it would be nice to have a range of products which are very different in terms of what kind of functionality you are looking for. So yes, we are definitely open for it. Yeah.
Yeah, and this is one of the facts that, I mean, we know building a consortium that each partner who comes in will have a sort of need to have inside of the consortium for it to be useful for them, and we're open to that. So if a partner comes in who says, well, we really want to look at, I don't know, bakery fillings, okay, yeah, that's part of the co-creation process. Thank you, Martin. Good question. Let's see. Annabelle also has a question on the scope of what types of plant proteins we'll be using. Would they be native plant proteins or treated industrial? I guess I'm reading that into it. Annabelle, if you want to hop on, you're welcome to clarify if I've got that wrong.
I think our aim was to also make the work relevant, was to actually use commercially available plant protein, ideally coming from our partner, for a few reasons. I think the processing of a plant protein towards an ingredient has an impact on its functionality. So yes, you can do it with native plant protein, but how relevant would it be for our industrial partner? It's, at least for me, would be the question mark to actually only work with commercially available plant protein.
Okay, I've lost track of the order of things. Let's go over to the people who very politely raise their hands while I read the rest of what's in the chat. Monica, you're the first one raising a hand.
Yes, thank you for this nice presentation. Super interesting project. And I have also a question related to the plant protein, maybe to be a bit more specific on this, what kind of plant proteins you use. Will you have some kind of a matrix where you, for example, say, okay, in the end, I will want to have like a heat gelled food. And this is why I selected plant protein that has similar thermal properties than our recombinant protein has, or the end product will be based on an acid-induced gel. So this is why I only look at plant proteins that have similar properties. So will there be, I mean, only a selection coming from the partners, or will also be the very systematic approach in the beginning while selecting it? Or, for example, if you want to have a stretchable cheese, then you would maybe select the plant proteins that is also stretchable. So something like this.
Very good question, thanks. What I would have in mind, and Julia, please correct me if you disagree, but is that on the beginning of the project, we would have an activities which is on the scoping of the type of ingredient that we bring. So no, we will not limit ourselves to plant protein coming from the partner. Ideally, you would want a range of protein which are from with different functionality, but also not only legume and pulses, but it can also be cereal, depending on the type of applications that we are thinking of as well. So I would really like to have a range of proteins that we can test in to make the toolbox also in work package 2 more relevant, because if you want a matrix with different mixing strategy, I think you need to have a range of protein from different groups.
Thank you.
Yeah, welcome.
Laurice, maybe just to clarify in that, do you have thoughts on proteins that are available now, proteins that are available short term, maybe slightly future proteins, or long term, like wild proteins? Where are we drawing the line?
Yeah, so from existing protein, I would think of soy, pea, maybe soybean. As from cereal, you can think of wheat or oat protein, for example. And from the newcomer, if you want a highly functional protein, maybe Rubisco could be one. or you could even think of mycoprotein or single cell protein if it's of interest. But it's not a protein by itself. In this case, it's a microorganism. So I think together with a partner, we will need to set the boundaries either already in the proposal or on the beginning of the project together to create boundaries of protein of interest.
Yeah, thank you. I think the two questions in the chat should be answerable quickly, so we'll hop over to those and then come back to the raised hand. So Anushree asks, are we flexible in terms of working with non-dairy animal-free protein? So I could imagine egg or gelatin. Are we open to doing that if that turns out to be a priority for one or more partners expressing interests?
It's a good question, and I
Yeah,
don't really know the answer.
Yeah.
With the title of the project and the focus of the project, my first instinct would say no. But maybe I'm not saying it's not interesting because it is. But for the scope of the project and the focus of the project, I would say no.
Yeah. Yeah, I think that, I mean, we'd like to hear from all of you, as I started in the beginning saying, the project doesn't have to be exactly as we envisioned it. We've envisioned this as a dairy-focused project, but if there's a critical mass of 3 or 4 companies who say, hey, we really want to do this for egg, I'd like to know that, and then we can have a conversation about whether it's possible to put a work package in that's looking at that. If it's only one partner who's interested in something, it becomes difficult to be able to finance the level of effort that we need to answer that. Okay, let me do Dilek's question, and then we'll take the hands again. Are we going to do technoeconomics? Is that in scope to do techno-economic models and cost and use calculations?
It's not something that we included in the slide, but it's something that I could see fit into the project, and also for the funding agency, I think it might be relevant to do a sustainability evaluation. So cost and sustainability evaluation. So yes, I would say yes.
Yeah, great. Thank you for the question, Dilek. Okay, Weza. Your hand raised.
Sorry. Thank you for the presentation. May I ask, will you consider not the precise fermentation of the protein, but it's, as you mentioned, it's like single cell protein, like, for example, the yeast protein, the microbial protein?
So what you are asking is just using the yeast, right?
Yeah, yeast protein.
Yeast-derived protein.
Yeast-derived, no, the whole...
Not the whole. We already have the product that has been extracted the protein from the yeast.
Okay.
I mean, not the body itself, but the protein. For example, the protein content has over 80%.
Mm-hmm. At least for me, I would not see it as a replacement of the dairy, but it would be a counterpart of it. So does this protein or this extract, does it has food grade approval, or is it food grade?
Yes, for sure.
Okay.
Yeah.
So in principle, it would be the counterpart of the animal-free dairy protein. It could be one of the proteins that we try to mix with the precision fermentation protein. Yes.
Oh, okay. Okay. Thank you.
Yeah.
Yeah. So that's the wonky. I mean, we label it plant-based because that's one of the protein. I mean, that's the category. Yeah. But some of these single cell proteins are actually interesting for us as part of exploring plant-based. But we just don't have the right language to capture plant-based and microbial, or else the project titles get way too long. But thank you for the question to clarify on that. So I'm going to answer Monica. Yeah, great. I mean, Shiru is a partner on one of our public-private partnerships started in a previous round. I think they're great and doing amazing work. Great suggestion to reach out to them. I would love to see them coming on board And because what they're doing is so interesting, we can try to accommodate how they can be a potential knowledge partner. Thank you for the suggestion. I will reach out to them. And feel free, I love that when we have partners on board who see another complementarity, you're all welcome to recruit your own partners to our project. It makes our lives easier, in fact. So love those suggestions. Please keep them coming. Paul, thank you for raising your hand.
Thank you, Stacy, and thank you, Loris, for the good presentations. Maybe my question, I have two questions, and then on the cost in use, I think it's essential to include this in the project. Definitely, especially when you are trying to make an application on a certain protein content, depending on the purity, you have to consider cost in use. So I think it's an essential parameter. Maybe two things. One, you are looking at interactions between proteins, but there are many products on the market, thinking cheese or yogurt, that may have other texturants like starches. Do we consider other texturants in the project, or do we say, no, we are just only going to look at the protein-protein interaction and see if we can find some synergies or no synergies in there? So that's one question. And then the second question is, there is one promise of these biotech ingredients, I call them, or more, which is reduction on LCA. What about LCA calculations of these blends?
Okay, first, the first question, the texturant. I think in WUR package 2, I think we would prefer to look at protein interaction mainly, and to see how do they performed. But in the model, so in WUR package 3, so in the application in model food products, there's other type of texturant will enter in account, so can be included in the formulation, and we can see how do the blend of protein behave and interact towards this other minor component. Yes. Yeah. So this is included for the LCA. And by what
CO2 footprint calculations.
Yeah. This is something. Yes, I see as part of the sustainability. So it's cost in use and the greenhouse gas emission. So of this carbon footprint that we can include in the project. Yeah.
Yeah, I think the question there is, do we do a complete LCA on all the materials, or do we have some shortcut methods to just assess sort of change parameters, whether we do a complete LCA or do something? But the subsidy agency likes to see us doing that proof that what we're doing is contributing positively to emissions reductions, for example. Yeah.
Especially if you have different levels of purity.
Yeah.
Which would require more processing and then impact the CO2 reduction.
Yeah, good point.
Yeah,
thank you for that.
There I would like to also mention, this will be my last comment, and that we also need a partner there to be on board for the LCA and the calculation, because we need information from you guys, and we can do it on a confidential basis to some extent, on the cost of production or the LCA greenhouse gas emission of your process from the plant, but also from the precision fermentation protein. To make it a really valuable result, it's very important.
So in view of time, what I'm going to do is give you the last message and say goodbye to everybody who needs to hop off because they have something else. And then Dario has put a really great question. So we'll stick 5 minutes for those who are able to stay and take whatever last questions there are. But just to wrap up, for those who have to go, we would love to have your feedback, whether you're interested in being a partner on one of these projects, or interested in working with us in another way. Or if your message is no, completely not relevant for us, you guys are totally on the wrong track, please, all of that feedback is very welcome and helpful. I'm here first because you can contact me for any of this. I'm sort of overseeing our work in this domain. But if you have questions for either of the two specific projects that that Paul and Laurice presented, please also feel free to drop them a line. Or if you want to talk to Mark or Martin, contact me. I'm happy to forward you on. What we're going to be doing in the coming weeks is some deep dive conversations with partners who show an interest in one of these projects to start that co-creation process. So love to have your, oh great, we've already got someone volunteering in the chat. Thank you so much for that. You can also volunteer in the chat to engage with us. I love that, even easier than my email. Thanks so much. And then, well, goodbye to those who need to go. I'm going to read out Dario's question because I think it's a relevant one. And if anybody else wants to stay and listen in, this will also be on the recording, so you can hop on and watch the last couple of minutes later if you want. Dario's question, how would sensory be handled in work package 3, given the regulatory status of some proteins involved? I'm going to give the first answer to this one, but Laurice, jump in if you want. So we would love it if we have partners who are in geographic locations where these products are possible for tasting. That's a good reason for us to approach and to proactively look for international partners. I will also say there's a development within the Netherlands. So last year, our Ministry of Agriculture created a sort of sandbox protocol so that companies and interested partners were allowed to taste cultured meat products. And they are intending to branch that out to precision fermentation derived components. So there is a very we have an optimistic outlook that within the next year or two, there will be a carve out also for tasting precision fermentation products in the Netherlands. That's not there now, but we can anticipate that that will happen in the scope of this project. But again, ideally, we would love it if we have partners who are able to perform that in their geographies. Laurice, is there anything you want to add on that?
No, I think it was clear. Yeah.
Okay, awesome. I think the rest of what I'm seeing in the chat is thank you and goodbye. So if there are any last questions, feel free to raise your hand now. Yeah, Sunil, our favorite question back.
Yeah, before I jump off, how open are you to jointly champion this for launch of, say, whey protein products in Europe as a whole?
Yeah,
and
shall I
and
take that
navigate
question?
through the regulatory maze.
Yeah. So we're very open for participation in discussions toward the European food safety authorities. We participate in multiples of those. We even, just to take an analogous sector, not that this is the one we're talking about here, but for insects, for example, we were part of a commission that was proactively engaging with the EFSA, and we supported a couple of the dossiers that were there, and we helped sort of facilitate that conversation. I think we're very open to it. That's not usually something we do inside of a particular project, but that's an activity that we do participate in as Wageningen, with our specific role being to represent the science. We are a science-based organization, so we don't lobby. We're not a lobbying organization. We're there to share the science and to help, help bring our understanding and make that available for our regulatory parties. We're also very connected to the Dutch Ministry of Agriculture, for example, on that sandboxing work. We're supporting and helping them to find their way as much as possible. Yeah. Thank you for the question. Sarah?
Yeah, I guess a question on the Profs project. It sounded like open to partners who could bring in kind of their own ferments to kind of put into this process. Are you guys also open to receiving kind of for work product 3, like things post downstream processing that could go into kind of an allergenicity test only?
Short answer, yes. Mark, do you want to jump in and give a longer answer?
Yeah, so I missed part of the question, but what exactly did you want to bring in?
Oh, if we could bring in things actually post DSP as well as things pre DSP.
Yeah, I think so. Yeah, sure.
Yeah. Consensus.
Yeah.
Thank you for the question. Any last thoughts or questions from the group? If not, then thank you all so much for joining. Also, thank you for sticking with us these last few minutes. look forward to hearing from all of you. I will send around the slides, also a link to the recording. You're welcome to share it with colleagues. And well, please reach out. We look forward to having your feedback. Thanks so much.
Okay, bye.
Thank you. Bye.
Bye-bye.
Bye, everybody.